Since 2014, more than 20, 30 new drugs are coming on the market every year, which is unbelievable for what we were accustomed to do. When I started to work, we were dealing with 5, 10 drugs, and now we have hundreds of drugs. So essentially, the new ones are mainly biological. In this moment, what is dealing the research and also the care is the immunotherapy, the new immunoagents. But nevertheless, there are still a lot of space for the target agent. The difference is that target agents are drugs that are very specific for some kind of molecular abnormalities in the tumor, and usually they are very active. Unfortunately, until this moment, they're not curing definitely any patient, beside a few cases. But we are able, with the research, to develop always new drug against different pathways of resistance to such targets. And this is very interesting because this is prolonging the life and improving the quality of life of our patient even with metastatic disease for years. As a matter of fact, we multiply by a factor 3 or 4 the overall survival of cancer like colorectal or lung and melanoma, and now we are starting with the gastric and so on, which is very good. But and then the other group of biological are the immunoagents. These new immunoagents are drugs that are dealing with our capability to renew tumor cells with our immune system that is somehow inhibited by the cells themselves with the physiological way that we have to defend from autoimmunity. Now, once that we discovered this, we were able to create a monoclonal antibody that are with the capability to reactivate the T cells activity. So the T cells are those cells that are active after that there's been an immune reaction against something from our body. In this way, we are obtaining extraordinary results in at least the majority of cancer we're treating, not in the majority of patients, but in the majority of cancer. The best result I've been obtaining up to now in melanoma, those tumors that have a macrosatellite instability, head and neck cancer, lung cancer, especially the squamous, and the renal, and those are already commercially available drugs for this disease everywhere in the world, anal, and so on, so many. What is the difference? Is that activating immunity, we hope that we are going to cure patients, definitely. And this is the only way, on my point of view, to improve what we call the economic toxicity of this new agent. Because as everyone is aware that these new drugs are very expensive and are particularly one of the major problems is the sustainability for the social security system, like in Italy, where everything is covered by the social security. But of course, prolonging the treatment for years at costs that are 10, 20 times higher than what we were accustomed to pay for a treatment is going to be really a great problem. The solution is to cure more people, and what we are doing now is try to understand better, since the beginning, how is the characteristic of the tumor and the host. So we are trying to get much more information we can, as much information we can from the biology of the tumor since the beginning to understand if we can improve the cure rate instead of the care rate. So transform what is activity and prolongation of survival in cure, which is, this is going to be the goal of the next few years. The second goal is going to be to understand if we have to prolong treatment like we're doing now, if it's enough to perform a boost of treatment at the beginning and so that we obtain a response, maybe to maintain the results with some kind of recall done once in a while, not instead of every month or every 3 months like we're doing now. Some of them must be used in combination with standard chemotherapy. I'm still strongly believing that chemotherapy is not going to die, because, as I told you before, all these new agents are curing a percentage of patients. Unfortunately, target agents are very effective on those tumors that have the target, and targets are in 3%, 5%, 10%, when we are lucky, 15% of a kind of tumor. That means that the majority of patients usually may have a tumor without a target, or if we put all the target together, at least half of the patient, from half to 75% of the patient, have not a biological cure with a target agent. Immunity seems to be more active, widespread, but even with immunity, we have a very different result in different patient populations. So even there, we should consider that we should select those patients that have a higher benefit and understand why it's not working. As a matter of fact, we are developing, as I told you before, my major effort is in phase 1, so in new drug development. And what we are developing now in many studies is the combination of the new immunological agents, combination between immunological agent and combination between agents and chemotherapy, agents and the target therapy, and so on. What is clear up to now, if there is a target driving the tumor growth, immunotherapy doesn't work very much. So for those patients with lung cancer that have a mutation of EGFR or rearrangement ALK or ROS1, something like that, the results are really disappointing. So for those patients, chemotherapy is better after the target therapy has failed. Or we are dealing with new target agents that are bypassing resistance. In all the other patients, immunotherapy can work, especially if there is a high number of mutations inside of the tumor that seems to be more immunogenic.